So thanks for the nice, kind introduction. My name is Corentin Bisot. So I'm a researcher, right? So I will talk mostly from my point of view of researcher, try to give you some insights from what working with living organisms means when you're a researcher, and also what you find when you look at them. But since the scope of this talk is also to go a bit beyond what the simple science, I will also try to give you some more general insights with limited legitimacy for it. So I will tell you when I'm going beyond my own standpoint as a researcher. So as a researcher, I'm interested mostly in microbes, and I'm interested in morphogenesis. So morphogenesis comes from, I guess, Latin or Greek, something like this. And it's about making a shape, right? So it's how a shape grows from, an initial seed, let's say. And it turns out that when you think about morphogenesis, you're thinking about a final shape. And this is very true when you're thinking about a cat, right? About like some very nicely organized animals. But if you go to the microbial world, things start to act a bit different, right? You're not really achieving a shape. Collective of cells still come together, but they are constantly moving, and it's at the same time a shape and a behavior, as you will see. So that happens at the level of bacteria, right? Bacteria often come together and form collectives. We talked about biofilm a little bit earlier. I will tell you a bit about slug forming slime molds, so this is maybe a bit cryptic for now, but I will tell you a bit. And just to give you an example that sometimes living things are a bit in between, if you think of a tree, for example, it has flowers that are really well organized, right? If you look at the flowers, all the flowers of this Magnolia tree are organized in the same shapes. They will have the same potentially amounts of petals, I guess. I'm not sure how you say that in English. But the tree itself, right, the trunk and how it branches, etc., is going to be every time different depending on where the tree grows, because it has had to adapt to its environment, and so the tree itself doesn't have a well-defined shape. So somehow plants are a bit in between these two words, macroscopic words of like, adopting a behavior and responding to your environment, and development where you have this cat which has a very final shape. When thinking about living things, I also like to introduce these 6 letters, which I really like, and you will find out about them in the workshop as well. And they're basically atoms, right? So don't be scared. They're just the letters that allow you to write everything that's alive, right? So everything that's alive is made out of those 6 letters, and everything that's alive needs those 6 letters to survive and grow and divide, which is what living things do. You may recognize some of them, right? Carbon, we already talked about. O is oxygen, that's what you get in CO2, but H is hydrogen, and that's what you get in H2O, which is water. And a lot of these elements actually have an origin in the soil. And we as humans are also living things, right? So we need also those elements in order to survive and grow. And one method we found in order to provide them to ourselves is to grow plants. And then we can transform those plants into baguette, which was mentioned also earlier, and eat them. In order to grow those plants, though, we need to modify the environment. So we need to provide those plants with what they are missing. And photosynthesis was also mentioned earlier, right? Plants are really good at this. They're really good. They're like the power, the energy lord in the land. But they're, the underground is a bit of a chaotic system to find nutrients in. So they also need nutrients to grow and survive. Nutrients being the nitrogen and the phosphorus. So what we do in modern agriculture is add those nutrients to the soil. And this is nice, the plants grow, we eat, and we're happy. But those nutrients also leach out in the river, in the waterways, and these end up in the sea. And what you see here is an example of an algae bloom, right? So it's bluish, like clear blue. So these are algae that are very happy because there are lots of nutrients, and because they have access to energy, because they can photosynthesize, they just grow a lot, and that's very nice for them. The problem being that they also need oxygen. I mentioned it earlier, and because they need oxygen, and oxygen is only present in a limited amount in the ocean, they deplete the oxygen stock in the ocean, which leads to less oxygen for all the living organisms, and this is an extreme case where fish can die because of it. So my point here is that we're embedded in those cycles. We modify them. We keep we need those cycles in order to survive, but we're also modifying them. And a lot of them essentially originate in the soil. So if you look at where these arrows were coming from, they all came from the soil. And this is because the soil is a very complex system. It's basically a bunch of particles on top of each other, but it's unlike Tetris, they never fit perfectly into each other, so there's always space. And in this space, you can have water, you can have air, and you can have living things that move around and die and poop, and that's what makes a soil. I already mentioned I'm interested in soil, and in my research I study mainly two organisms. One is Mycelium discoideum. So it's a slime mold, and you see here a video of it eating bacteria. You can see it's always like moving around, crawling around like this. It's a very funny cell. It looks a bit like the lymphocyte that you have in your body that are your immune system basically, but this lives alone in the soil. So it's a single cell organism and a bacterial predator, but it has the ability to switch between single cell and multicellular states. So this is another video which I took in the lab, and I will stop it because I need to present to you what's going on first. Okay, well, you get a spoiler. It's really hard to control. Okay, so all the purple dots you see here are the same cell you see on the left, but just zoomed out, right? So you have a lot of purple dots. And when I play the video, you will see them moving around. So they are in the single cell state, and then you will see that after a while, they come together, they form this big group of cells, which is bright purple, and then moves around as one. So that's one of the things you find in the soil. The other thing that I'm interested in, and I think that was how the talk was branded, so I'm not going to talk too long about slime mold, even though they're cool, are mycorrhizal fungi. So the principle is also that they are embedded in the same cycle, right? So the plant is photosynthesizing, right? It has a lot of carbon, it has a lot of energy available, but it's looking for nutrients in the soil. So the plant will give nutrients to a growing fungus, which will extend its mycelium, so its network of hyphae, into the soil. This will find phosphorus and nitrogen, so all the nutrients, some of the nutrients necessary for life, and will provide them to the plant. So it's a win-win situation, right? Both of the partners are getting something. The fungus is getting energy and carbon from the plant, and in exchange, the plant is getting nutrients. The ones I study are called abuscular because within the plant cells, there is this very intimate relationship, and they really form actually, well, fungal cells inside the plant cells. So what you see here is a really zoomed in version. It's not the outside mycelium, which is in the soil. It's really what's inside the cells. They're super widespread, right? 60% of plants on Earth associate with this kind of fungi. And they've been around for more than 400 million years. So it turns out that people think that when plants arrived on land, they didn't really have a root system. They basically like just algae. And they associated with those fungi in order to have some kind of root system. So I've been studying in the last 5 years those fungi in the lab. And to study them in the lab, you have to get them out of the soil because the soil is very nice, but it has a big disadvantage of being opaque, so you cannot see what's going on in there. You have indirect ways of sampling what's in there, but you don't see it. So the technique is you put them in a petri dish, right? So a petri dish is what's usually used in biology in order to grow microorganisms. So it's a bit of a special one because it's split in two. In the bottom half, the brownish thing you see is a root. It's a bit of a weird root because it doesn't do photosynthesis, so it's a bit of a zombie. It's actually eats sugar from the agar, so from the medium. Despite it being a weird root, it's still alive and it's still able to form symbiosis with a fungus. So we put them together in this bottom half, but there's a thin mesh that separates the two halves, for which only the fungal hyphae, so the filaments that compose the body of the mycelium, for which only the fungal hyphae can pass. And when they pass, we start imaging them in a robot, and we take image every 2 hours, so we get the kind of time lapse you see here. So this one is not moving, but you will see a lot of them later. So again, this is called a hyphae, and yeah, this is basically the robots we have in Amsterdam in order to image them. So a lot of them have been built by Loreto Ugarte Galvez and the team of engineers there. So it's really advanced. Well, this is a bit too fancy in my opinion, but still it works. And that's how we acquire many movies of this. so in the end, we have terabytes of data of this fungi growing, and I've put a little music here. I don't know if it's going to be loud enough for everyone, but I will leave it. So I have actually a 5 hours movie of this, but it's only the 2 first minutes I'm going to put here. And I'll let the music just for 1 minute so you can meditate a bit. And I'll have you look at it and try to think about one question. Ooh. This is a new thing. Should I type this? Okay, unfortunately I cannot play the 2 full minutes, although it's very charming. But I can share the movie if you want to watch it at home. Here's a different thing where we grew it from spores, so you see the spores germinating. All right. Oh yeah, this is contamination. That's a fun one. So it's another fungus that just happened in there and started overgrowing the one we wanted to see growing. So it looks like a big, I don't know, atomic bomb or something. All right, sorry for interrupting the, the music. I feel like my voice is awful now. Okay, so this is one of the movie I selected, and here I highlighted in different colors all the hyphae, and the color here corresponds to their time of appearance. So basically you're seeing a movie, but you're seeing it twice, because in the final image, if you just look at the colors, you see when each element appeared. So now I asked you a question before you watch the movie, so if you didn't fall asleep, did you have any answer to my question? The question was, how do you pass from one single hypha to a full network of filaments? Okay, I see nobody raising their hands, it's nice. So that means I really, did research and found something that's not obvious. So what we eventually found, and we think is true and actually goes beyond only this fungal network, is that how you go from one single filament to the full network, which is then able to perform all its function, acquiring nutrients, etc., is just based on 4 simple rules. So every single filament can do essentially 4 different things. It can grow, so get longer. It can stop, so that means it will stop expanding and then just, stop from there. It can branch, so then a new filament will emerge from it at a given angle. Or it can merge, so when it finds another filament in the near vicinity, they'll come together and make, a fusion, and then essentially the whole network you see here is one cell somehow. I mean, everything is connected in there, so it's an open pipe system. Things can flow through it, so then it really merge together. So if you take those rules and check whether they actually happen, we find that it seems to be the case, and you can then implement those rules in a synthetic network. So this is what you're going to see here is a simulation. So I use the same color coding, but what you see here is that you get something that looks like a fungus. Is maybe not as elegant as the actual one, so we're probably missing a lot of things, but in terms of how it grows and how it expands in space, you're capturing some essential elements of it. I'm realizing I'm already a bit long, but I'm good. Okay. So this leads me to my take on cellular collective and aesthetics. And here I go, away from my research. I'm just going to give my own subjective note on it. And what you're going to see here on the left is basically the same. It should appear eventually. No? Okay. It's basically the same model, right? With filaments that are growing, branching, maybe fusing, maybe stopping, but this time implemented in 3D. So that's actually more like what it looks like probably in the soil. And what you see here on the right is a monkey. And I already told you about my own feeling about the difference between these animal organisms and these networks or these microbial collectives. I think we're very used to these kind of shapes, and we're very used to the kind of symmetry, to the kind of beauty we find in them. Maybe a bit less to the kind of beauty you would find in microbial collectives, despite them having, not so well-defined shapes, but very nice, maybe dynamics. It's all a subjective matter, but I really like the movie I just showed you, and I also like these simulations. I think, although them lacking some symmetry and lacking maybe some essential elements of order, as you would see in the monkey picture, they are still very pretty. So that's one slide about cellular collective and aesthetics. Then I have one slide about cellular collective and ethics, and I will play again this movie that you saw before, and I will tell you a bit more what I was trying to do there. So my research currently is on these slime mold cells, right? Remember, bacterial predators, so they eat bacteria. And what I do is that I put a lot of bacteria in the top thing, right? So this has a lot of food, and this doesn't have a lot of food. And in between is an air gap. So it's really like, for microbes, some wall that is really hard to cross, at least on your own. And what we find in many of these experiments is actually that by coming together, so it's a bit of a very nice story. They're coming together and they're able to bridge that wall and cross across the air. So it sounds like getting together is very nice, and we should get together, etc. But the other thing you see in that movie and in this final picture is this, little purple thing, which are cells left behind. And it turns out that for cellular collectives, especially the ones that are completely sharing the same DNA, they really don't care about having 90% of them dying. They just need their own DNA to be propagated and being able to go to the other side. So in that sense, I don't think there is really any ethics to derive from microbial collectives, except a very weird living one, which consists in just surviving. And that might be my conclusion, which is that it's almost odd to speak about design and biology, because biological beings are by essence not design, right? They just happen. There's just evolution that's driving them to emerge at some point. And in that sense, the only conditions of them existing is them to be able to exist and survive and divide. And I like this picture of one of the pristine forests that's still there in Europe, right? It has existed for thousands of years, so we're moving from the picture of a microbial collective to the picture of a collective of many different organisms. And I would say this forest is a mess, right? There's like moss on fallen trees, there's like trees all over the place. Probably if you would look in the ground, it would be even more of a mess. There's like earthworms moving in completely erratic directions, right? But despite this, it has existed and survived through thousands of years. So in some way, it's able to survive and have, at least some of the ecological function that it's able to do. And I think in an era where there might be a temptation for frozen order, maybe getting inspiration from how messy living things can be and still how functional they are might be some good thing to think about. Thank you very much.